Alright guys, so today we're going to be talking about photosynthesis. You've been doing the reading on photosynthesis and hopefully you've drawn a diagram of what you know about photosynthesis. Now we're going to look at some specifics about photosynthesis and take a few notes based on what you read. So let's talk about just some basics. First off, what is photosynthesis? Photosynthesis is a process in which light energy is captured in is captured as chemical energy for all living organisms. Now, here's the deal. We said all living organisms. Now, all living organisms do not capture light energy and convert it to chemical energy, but all energy available for all living things originates here during photosynthesis. It can't be used by living organisms unless it is captured through the process of photosynthesis from light energy. So photosynthesis provides all available chemical energy for all living things on the planet. It also is the only way that we have a way to incorporate carbon into the organic molecules that we need for life. Carbohydrates, lipids, proteins, and nucleic acids, the only way we get that carbon is through the carbon being captured from carbon dioxide and captured into glucose through photosynthesis. What type of organisms do photosynthesis? Autotrophs, meaning they make their own food. We can also call them producers. So this would be plants, some bacteria, and some protists. Where does it occur? It occurs in the chlorophyll and the chloroplast. But if you notice, we put some bacteria can do photosynthesis, and bacteria are prokaryotes. Prokaryotes mean they don't have membrane-bound organelles, meaning they don't have chloroplast, but they do have what we have inside of a chloroplast called chlorophyll. It's the pigment that makes plants look green, but that pigment allows for the capture of light energy and the conversion of that into chemical energy. The overall reactants, light energy, carbon dioxide, and water. And the overall products, we get oxygen, and we get glucose, which is C6H12O6. So this is just big picture, what's made. Now, oxygen is a byproduct. It is not something that the plant, it's not the purpose of photosynthesis. The purpose of photosynthesis is to capture energy in the form of glucose so that energy can be harnessed and used by a living organism. But oxygen is created by a byproduct, and we do need oxygen. It is really important. So let's talk about some basic steps. There are two basic stages of photosynthesis. And the first stage is the light dependent reaction. And what happens in this is that light energy is absorbed, water molecules are split, and oxygen is released. And the last thing is energy is transferred. Now, guys, big E means energy. So energy is transferred as ATP and NADPH. Now, if you look at our overall reactants, we have to have light energy, carbon dioxide, and water, and we produce oxygen and glucose. So let's look at the light-dependent reaction. In the light-dependent reaction, we use the light energy, and we use the water, and we produce the oxygen. So that's the first step. In the second step, guess what we don't need? We don't need light. So this is called the light independent reaction. It's also known as the Calvin cycle. Now in a minute we're going to watch a little short video, and it talks about the Calvin cycle, but it also calls it the dark reaction. We're not going to call it the dark reaction because that gives the idea that it can only happen at night, and that's not the case. The Calvin cycle can happen in the day. It just does not depend on light. It is independent of light energy, because what happens is energy from the light dependent reaction is used with carbon dioxide to produce glucose. So now let's look back over here at our reactants. We're using the carbon dioxide, and we are producing the glucose. Our main purpose is to produce that glucose. Where did this light energy go? Well, it traveled from the sun through ATP and NADPH, and it ended up in the glucose. And that is how that energy gets into the food that we eat, is through the energy being captured and stored into glucose. Now, a couple other things about this: the light dependent reaction happens in the thylakoid of the chloroplast. And the light independent reaction happens in the stroma of the chloroplast. We're going to watch a little video about photosynthesis. It's short, it's about 2 minutes. It just goes over the basics, and then we're going to look at a diagram of photosynthesis and fill it in and make sure that we understand the basics of photosynthesis. That's all we're trying to get this semester, is the basics, each of the types of reactions, what goes in, what comes out, where it occurs. So go ahead and watch this.
The leaves of this plant have cells that carry out photosynthesis. During photosynthesis, plants capture the energy from sunlight and use it, along with water and carbon dioxide, to produce carbohydrates, or sugars. Oxygen is also released as a byproduct. These photosynthetic reactions are typically written as a chemical formula: solar energy plus 6 carbon dioxide molecules plus 6 water molecules react to produce 1 glucose molecule and 6 molecules of oxygen. To better understand the reactions of photosynthesis, let's take a closer look at the leaves of this plant. If we zoom in on one photosynthetic plant cell, we can see the chloroplast, where the reactions of photosynthesis occur. Let's zoom into this chloroplast to get a closer look. Photosynthesis consists of 2 primary steps: the light reactions and the Calvin cycle reactions. Both steps occur within the chloroplast. The light reactions occur within the thylakoid of the chloroplast. Here, special pigments absorb light energy and transfer it to high-energy electrons, which ultimately produce ATP and the electron carrier NADPH. Water is also split to replace the high-energy electrons. In the process, oxygen gas is released. The Calvin cycle reactions, commonly called the dark reactions or the light independent reactions, occur within the stroma of the chloroplast. Here, A series of reactions use the ATP and NADPH produced by the light reactions to turn carbon dioxide into carbohydrates. The NADP plus and ADP that were also produced by the Kelvin cycle reactions are then recycled to be used by the next set of light reactions. Now that you understand a little about light reactions and Kelvin cycle reactions, let's zoom back out and do a quick review. During photosynthesis, plants capture the energy from sunlight and use it, along with water and carbon dioxide, to produce carbohydrates, or sugars. Oxygen is also released as a byproduct. These photosynthetic reactions are typically written as a chemical formula. Solar energy plus 6 carbon dioxide molecules plus 6 water molecules react to produce 1 glucose molecule and 6 molecules of oxygen.
All right, guys, so let's look at this diagram. This is a great tool to study. In fact, we will take a quiz tomorrow that will cover this diagram. It will be this diagram and the equation for photosynthesis. So let's go through this. Let's talk about it. Let's talk about why this diagram is so great and all the information you can get from it. But let's start off. Photosynthesis, like we said, it's the process in which autotrophs capture light energy and store it as chemical energy so that it can be utilized by organisms, and it stores it in the form of glucose, which can eventually be stored as starch. Now, the equation for photosynthesis is 6CO2 plus 6H2O plus light energy. Now, this light energy, it can be written above the arrow or as part of the reactants. Either way is fine. And we produce C6H12O6, which is glucose, and 6 oxygen molecules. So there's our equation. Know the equation. You will have to know the equation on the quiz, but memorize this equation because it makes things so much easier, and I'll show you more about that in just a second. So first off, this is a diagram of the chloroplast. And we need to understand a little bit about the structure of a chloroplast. First of all, chloroplasts are in eukaryotic organisms. They're in eukaryotic cells, specifically plant cells, autotrophs. And the chloroplast, it has a couple of structures you need to know. And if you look at the diagram, you can see how there are these stacks of discs. Well, these stacks of discs are called grana, or granum is plural. And each of these individual discs here is a thylakoid. And each thylakoid contains chlorophyll, that pigment that makes the chloroplast look green and makes a plant look green. It's inside each of those thylakoid discs. Now, the empty area around all of these grana, the empty area is called the stroma. And the stroma, it's literally the inside of the chloroplast is filled with like a gel-like material, kind of like cytoplasm. Well, that gel-like material is called the stroma, and it is where some of the reactions for photosynthesis actually occur. So in the first part of photosynthesis, we need light energy, and we need 6 water molecules. And we know these are water molecules. You can see the Mickey Mouse shape, and there are 6 of them. That does say water. H2O. And notice the arrows are going into the chloroplast. That tells us that it is our reactants, and the reactants are entering into the reaction, and as they'll go into the thylakoid, and through the process of the light-dependent reaction, we will produce 6 O2 molecules. We will also produce NADPH, and ATP. This is the energy transfer. the transfer of that light energy to the next step. The next step does occur in the stroma, and that is the Calvin cycle, or the light independent reaction. And in the light independent reaction, we use the energy that was captured in the light dependent reaction, as well as 6 CO2 molecules, and we produce glucose. C6H12O6. All right, so let's look at this diagram for just a second, so maybe you can see why I think it's just so great. First of all, reactants are what we start with, products are what we end with. So let's look at our equation. Our reactants: carbon dioxide, water, and light energy. That's what we start with, and if you follow the arrow in the equation, they're going to go into the equation. So let's look at these arrows that point in. We have 6 CO2, 6 water, and light energy. So if you know the equation, you can fill the diagram, or if you know the diagram, you can give me the equation. Then our products, we get C6H12O6 and 6O2. They come out of the reaction. So follow the arrows out of the chloroplast and we get glucose and we get 6 oxygen. So I love the fact that it gives us the equation. It shows us where it goes in, but this also gives us the ability to look at each reaction. So we have the light dependent reaction and the light independent reaction, and we can see where it happens, we can see what happens. So light dependent, and it occurs in the thylakoid, and then we have the light independent, and it occurs in the stroma. So great diagram. Tomorrow your quiz is this diagram and the equation. Make sure you study it, make sure you're ready for it. But this is a great overview of photosynthesis. We're going to spend a little bit of time exploring more information about photosynthesis now and more about the pigments and how it actually works and how photosynthesis comes together to function in the plant. And to do that, firstly, we need to understand a little bit more about the chloroplast. Well, the chloroplast, like we said, is an organelle in a eukaryotic cell. And it has a few unique features. Those features, it has 2 membranes. It contains its own ribosomes. It contains its own DNA. And it replicates on its own through what we call binary fission. And this is how bacteria reproduce. Now, why am I telling you this? I'm telling you this for you to understand that the idea, the theory is that at one point, the chloroplast was a bacterial cell. It was a fully functioning prokaryotic cell that at one point was engulfed by a larger cell. So here's our chloroplast, here's our big cell, and what is believed to have happened is this big cell, through endocytosis, came and engulfed the chloroplast. Well, remember in endocytosis, when a large food particle is taken in, it's taken in inside of a vesicle. So once it was taken in inside the vesicle, it has 2 membranes. And once inside that vesicle, this bacterial cell began to just function and live inside of that cell, inside of that larger cell. And as it started to live inside of that cell, it started to benefit the larger cell to have that in there because it allowed it to capture energy from sunlight, and it didn't have to eat anymore. It didn't have to be a heterotroph. It was now functioning as an autotroph. And over time, we saw that chloroplast replicated in that larger organism or in that larger cell. And as that cell replicated, those cells then had chloroplast inside of it. This is the theory of endosymbiosis. Endo meaning inside, symbiosis meaning to live closely with. So we learned about symbiosis and mutualism and commensalism and parasitism. Well, endosymbiosis is where one organism lives inside of another. And so the theory is that at one point the chloroplast was its own organism that started to live inside of another cell. And this was the beginning of what we now see today as eukaryotic cells, because we started to see membrane-bound organelles. So those unique features allow us to understand a little bit more how the chloroplast can function and how that its own energy metabolism can be used to help support the eukaryotic cell. So now let's look at how the chloroplast and the plant cells come together to work in the leaf. But first, let's talk about some terms that we've come across in the reading and in the notes that you need to make sure that you're familiar with. Let's start with the simple one, photosynthesis. Thylakoid, stroma, chloroplast, chlorophyll, light dependent, I'm abbreviating, the light dependent reaction, light independent reaction, RXN, that is an abbreviation for the word reaction. Endosymbiosis, and I think that's a good start on the list. Now, as we go through the next few sets of questions and models, add to this. If there's a word you don't know or a word you need to make sure that you understand, add it to this list so that as you're studying, you can go back and say, check, understand that, check, understand that. Ooh, I don't understand that one. I better go read about it. So use this to help you. Just add to your vocabulary list as you go. All right, so you're going to complete the modeling, the photosynthesis modeling activity. It says pre-AP lesson, oh gosh, I'm going to tell you. Well, I can't find my paper for it right now. Maybe it's it. It says handout 3.10, model-based guided inquiry, photosynthesis overview, and it has a picture of the chloroplast, and in the chloroplast you can see the double membrane here. So hopefully you're looking at this page right now as we're going through this, but you can see the double membrane. These stacks of thylakoid are called the grana, each individual disc is a thylakoid, and inside of that thylakoid, that's where we find the chloroplast. The empty space around it is the stroma, and the stroma is like the cytoplasm. And if it was a cell at one point in time, it would make sense that it would be full of a jelly-like substance similar to the cytoplasm. Excuse me. Pause this video while you work through and answer these questions, and then when you are finished, unpause the video and go through your answers with me as we work through this. So go ahead and pause your video and work. When you're done, unpause the video and we'll move on. So let's first talk about questions 1 through 5. Those are ones that we have already addressed in the first part of this, but the reactants for the chemical reaction known as photosynthesis, that's carbon dioxide, water, and light energy. When photosynthesis is complete, the products that can leave the chloroplast are oxygen and glucose. The balanced equation is 6CO2 plus 6H2O plus light energy yields C6H12O6 plus 6O2. The energy source that allows photosynthesis to occur is, the sun. So now we're going to move on and we're going to actually look at the structure of a leaf and how this all comes together. And in the paper, it went over the term spongy mesophyll, palisade mesophyll, epidermis, vascular bundle, xylem, phloem, stoma, and guard cells. So I'm going to be using those terms as we go through this, so hopefully you've paused it and read these things. But if we look at the diagram of the leaf, this outer layer is called the epidermis, just like we call our skin the epidermis. It's that outer protective layer. Then we have the palisade mesophyll. Now meso means middle. So the palisade mesophyll is in the middle. It's between two layers of epidermis. Now palisade, when you read the description of the palisade mesophyll, it says the layer of cells is located near the top of the leaf and is characterized by tightly packed photosynthetic cells. Notice they are photosynthetic, they are green, the chloroplasts are lined around the outside because the central vacuole takes up most of the center space where the water is being stored. But I remember palisade mesophyll because you can see that these cells are like columns all tightly packed in, just like a palace would have lots of columns. So that's how I remember that. And then if you look at the second portion of it, still photosynthetic cells, lots of chloroplasts. In my picture, you can tell it's green. They are not as tightly packed, and there's air spaces, kind of like a sponge with all of its pores. So it is called the spongy mesophyll. And then on the bottom layer of the epidermis, there are these openings. These openings are the stoma, or the plural is stomata. And this allows for gas exchange. Then within the spongy mesophyll, we see these bundles of circular cells. They are called vascular bundles. Vascular bundles are literally bundles of veins of that can carry or transport materials from the stems to the leaves, from the roots to the leaves or from the leaves to the roots. And these vascular bundles are made out of two different types of tissue. There's xylem, which transports water from roots to shoots. Shoots being the leaves and the branches. And then there's phloem. Phloem transports food, notice the alliteration, phloem, food, from shoots to roots. And what is the food that it's going to transport? The food it's going to transport is glucose. All right, so now we've labeled our diagram, and the next few questions ask us to draw some arrows. So the first arrow says, draw an arrow indicating the pathway you think CO2 follows to enter the leaf. Well, in order for something to get into the leaf, it has to have an opening to get through. And we noticed that we talked about these pores, these stoma are openings on the bottom. Well, that is how things get in and out. That's how gases enter and leave the leaf. So CO2 would come through the stoma into the leaf. And the next question says, this is letter B, it says, through the process of transpiration, water is brought into a plant through its roots and carried to the leaves, from which it evaporates. Which structure brings water into the leaf from the roots? That's those vascular bundles, specifically the xylem, but the water is going to come into the leaf through those vascular bundles. Remember, it's coming from the roots into those leaves. And then when water evaporates out, remember, water evaporates out through a process called transpiration. When the sun hits that leaf, it causes water to heat up, and as water heats up, it evaporates out, and it will leave as a gas through the stoma. It can't just leave, it has to have an opening, so it leaves through the stoma through transpiration. And then D asks us to draw and label an arrow indicating the pathway that oxygen would follow. Remember, oxygen is going to be made in photosynthesis when water is split, and when oxygen is made, it has to leave the leaf somehow, and it has to leave through an opening. So the same way the carbon dioxide came in, the same way the water vapors can leave, that is how the oxygen can leave as well. So hopefully your diagram looks something like this. If it didn't, go ahead and adjust that. And that gets us all the way through question number 1. All right, so we're still looking at this drawing. Real quick, we'll draw this back. That CO2 can go in, O2 can come out, and H2O can leave. And it actually comes in through the vascular bundles. So if we look at question 3, it says, if the guard cells close the stomata, what molecules will be unable to enter the leaf? So now this has closed. So what's not coming in? Carbon dioxide. And what would happen to the rate of photosynthesis if the stomata were closed? Well, think about the equation for photosynthesis. Carbon dioxide is required. So if we're not letting any more carbon dioxide in, the rate for photosynthesis would decrease due to lack of CO2. And under what conditions would a plant close? Well, let's think about this. And you might say, well, maybe it has too much CO2 on the inside, possibly. Or maybe it's trying to keep oxygen. And I'm going to go ahead and tell you the plant isn't going to try and store oxygen within itself. It doesn't want to keep all that oxygen because it has an adverse effect that we'll talk about much later. Could it be because it doesn't want to lose water? Well, if you think about it, when a plant, when it's really dry, and we're going through a drought, plants can still stay alive. But if they don't, what happens to them? If there's not enough water, they die. So plants are going to try and conserve water any which way they can. So if there's a drought or very dry conditions or very hot conditions, that would cause lots of transpiration, then we're going to see these stomata close, and if they close, then water can't get out, and it reduces water loss. And then it says, what does the statement structure fits function mean? And provide an example to illustrate your answer. We've talked about this multiple times. We talked about the structure of the small intestine with all the villi and microvilli increasing surface area. We talked about the inner membrane of the mitochondria increasing surface area to produce ATP. We've talked about the structure of an enzyme being so specific to the substrate that it gives it a specific function. Well, if you look at the leaf, the structure is still very specific to its function. The spongy mesophyll, very, very important in gas exchange. So there are air pockets where gas can build up and come in and leave. If you look at the epidermis, very tightly packed cells that are going to help protect the cell and help prevent water loss. Also, look, the stomata, most of them are on the bottom. There's some stomata on the top, but most are on the bottom to prevent the stomata from being exposed to direct sunlight. If that opening is exposed to direct sunlight and there's water inside, it's going to increase the transpiration, increase the water loss. So we see a lot of plants have most of their stomata on the bottom of the leaf to reduce water loss. the main function of the cell of the leaf is photosynthesis. So if you think about a tree and you think about photosynthesis, you think, okay, green things do photosynthesis. Well, where is the tree green? At the top, on the leaves. That's where photosynthesis occurs. Um, that's where it's most exposed to the sun. So the leaf, look at all the photosynthetic cells in that leaf. It's primarily photosynthetic cells. The only ones that aren't are those epidermal cells. So the main function of the leaf is photosynthesis, and that leaf is very much structured to function for photosynthesis. Now, we said water is produced, or water is lost through transpiration. We also said oxygen is produced in photosynthesis and it gets out. But what else is made in photosynthesis? Glucose. Well, remember, glucose has to travel throughout the entire plant, so that the entire plant has food and has energy. So glucose can travel through that vascular bundle, through the phloem, and be carried from the shoots or the leaves to the roots. And that glucose gets carried to all the cells throughout the plant, so that there's food everywhere for all of the cells to function. And we've talked about number 8. I just kind of combined questions 6, 7, and 8 all together. Describe the shape of a leaf, broad and flat, how broad and flat fits its function. Well, a broader leaf would lose more water. We're going to see more broadleaf plants in areas where there's a more availability of water. And the thinner leaves we'll see in areas where there's not as much water, but it's flat because it allows for gas exchange. You're not going to see leaves that are super thick because we need that sun to be able to penetrate through most of those cells so that all those cells can be photosynthetic. If it's really thick, the light energy isn't going to be able to pass through but a couple of layers. So those inner cells wouldn't be able to be photosynthetic. And then it says, do you think all plant cells have chloroplasts? Well, the leaf cells do, but do all plant cells? Not necessarily. We teach that the plant cells have chloroplasts. Well, they do, but not all of them are photosynthetic. Not all of them have chloroplasts. Think about the trunk of a tree. There's no need for it to have a chloroplast. So the cells that have the chloroplast function in photosynthesis. All right, so let's move on and let's talk about light for a second. Visible light is what we call white light. It's the light we see when we look up at a light bulb in the classroom. That white light is made up of several different wavelengths of light. It's made up of what I was told was my best friend when I was in 6th grade in physical science, Roy G. Bibb. Red, orange, yellow, green, blue, indigo, and violet. That's the colors of visible light. Now, when we look up at the light and we see white, we are actually seeing red, orange, yellow, green, blue, indigo, and violet, but all of those together because white light is the presence of all colors or all wavelengths of light. And conversely, black is the absence of light. So think about when you walk into a room and somebody says, oh, it's pitch black. It's pitch black because there's absolutely no light in the room, so we see black. So when we see the color black, we're actually seeing no light reflected off of the counters or reflected off of whatever is black. All of the light is absorbed, so no light is reflected back, so we see black. When we see green, what we're seeing is the green light being reflected back to our eyes, so that's what we see. All of the other wavelengths are absorbed. So when we see plants and we say plants are green, well, the reason they're green is because we see green, because it's being reflected. So since plants are green and green's being reflected, green would be the least useful color light to a green plant. But some leaves are not green. Some of them may appear purple, like in question number 3 here. So if it appears purple, what color is reflecting back? Purple. The green's being absorbed. So when you see a color, the color you see is the color of light that's being reflected. So if you're wearing a pink shirt, pink is being reflected, or those wavelengths of light that create pink are being reflected, everything else is being absorbed. This is why during the summer they tell you if you're going to be outside in the sun to wear a light colored or white shirt because it reflects all of the different colors of light. If you wear a black shirt, you're going to absorb all of that light and all of that heat, and you're going to get hot much faster. All right, so that was the end of our lesson on Monday, but I'm going to keep going so we can carry on into Tuesday since my my microphone wasn't working. So that was the end of Monday. So if you were out Monday, that's what you missed. We're going to keep going with Tuesday now. And Tuesday you came in. This was your first 5. This graph was on the bottom of your sheet from Monday, but there were now questions to go with it. And it says, which color in the spectrum will be most visible to you? And they're talking about in this plant. So if you looked at this plant in the graph, which color would be the most visible? Well, it's the one that has the highest amount reflected, so that would be green. And then it says what percent of the energy is reflected by that color. So for green, 95% is reflected, which means that only 5% of the green light is absorbed by that plant. So then it says if every color with more than 50% of light energy reflected is visible to the human eye, is red light part of the mixture of visible colors and light reflected by chlorophyll? Well, red light, here's our 50% line. Red light is less than 50%. So in this case, with this plant, the red color is not going to be visible. We're not going to see the red light because too much of it is absorbed. Now, if you look at this graph, my next question for you is, which color light is the most beneficial for this plant and its purpose of photosynthesis? That's right, the blue one, because it only reflects 10%. That means 90% of it is absorbed, and that energy can be used to create glucose. So the more that is absorbed, the more useful that color is. The less that is absorbed, the more of that light energy is reflected, and the less useful it is. All right, so let's do a quick review. Pause the video and write down the equation for photosynthesis. And once you've written the equation, draw this diagram. And label as much as you can. Pause the video until you've completed it, and when you are done, you can unpause and check your answers. This is just practice for your quiz. All right, so let's go over the answers. The equation for photosynthesis: 6CO2 plus 6H2O plus light energy yields C6H12O6, or glucose, plus 6O2. Now let's look at our diagram. This is a diagram of a chloroplast. This area outside of the thylakoid is called the stroma. This stack of thylakoids is called a grana. And one individual little disc in that grana, that is a thylakoid. And it appears green. Because it contains chlorophyll. All right, now let's talk about the actual reaction. What goes into the thylakoid? One thing that goes in is light energy, and the other is water, 6 molecules of water, in fact. And those molecules go into the thylakoid for the light dependent reaction. And through the light dependent reaction, energy is transferred. And you may have written it as ATP. and NADPH. If you did, that's amazing that you remember that. Keep that up. And also, water is split and oxygen is released during the light-dependent reaction. That energy that's transferred travels to the Calvin cycle, also known as the light-dependent reaction. And that energy is combined with carbon dioxide to produce our final product, C6H12O6. And this is where the energy from the sun ended up. And that is our food. It is food for the plant. It is food for the organisms that eat it. It is in the form of glucose. Glucose can eventually be converted to starch or glycogen and stored until the energy is needed. Now, today we did a virtual lab, and if you're at home, you're not going to do the virtual lab, but I need you to understand the idea behind it. So you're going to watch this little video. There's no sound to it. Just watch the video, and while you're watching it, figure out, what am I testing? What's the independent variable? What am I measuring for results? What am I going to record to tell if my independent variable had an effect? What is my control? What's the control group? What am I using as my standard of comparison? What variables do I need to keep the same? What are the controlled variables? And if I had decided to change something different, what other factors could I have changed? All right, so if you watched this video, you saw that there were a couple of things changing. Well, one thing changing, but in multiple ways. So here we have 1,000 lumens. The lamp intensity is at 1,000 lumens. And while we're doing that, we're exposing this plant that's submerged in water to 1,000 lumens of light. And we're watching these bubbles or oxygen being produced, and we're counting the number of bubbles. Then we increase the lamp intensity, and we watch the number of bubbles that are being produced, and we count that number of bubbles. And then once again, we're going to adjust the lamp intensity to 3,000 lumens, and we're going to count the number of bubbles produced there. And if you notice, the rate of bubble production gets faster as we increase the light intensity. So our independent variable is the thing that we are changing. We are changing the light intensity. The dependent variables, what are we measuring for our results? We're measuring the rate of photosynthesis, but to do that, we are measuring the amount of oxygen produced, or in this case, the number of bubbles produced. Things that we are keeping the same, our controlled variables, would be the amount of water, the temperature of the water, the distance the lamp is from the plant, the size of the plant, the type of plant. All of those would be controlled variables. Now, our control group is what we are using as a standard of comparison. Well, a typical indoor light is about 1,000 lumens. So 1,000 lumens would be our standard, and now we're going to compare the rate of photosynthesis as we increase that intensity. So the independent variable, once again, is the light intensity. Dependent variable is the oxygen or bubbles produced. And the control group, what are we comparing it to, is our standard, 1,000 lumens. And then everything after that is what we're changing. This is the lab that we did in class today. If you are at home, I will post a link to a virtual lab, and I'll post the link to the actual lab document. You can use the lab document, but then just conduct the experiment using the virtual lab link and collect your data, graph your data, and then go ahead and answer the questions from there. I hope this has been helpful to you. If you weren't here, if you have questions, please feel free to email me or ask when you get back to class. I really don't mind helping at all. Photosynthesis can get confusing. This is what the lab document will look like, or is what it looks like. Hopefully you printed it out. But let's talk about factors that can affect the rate of photosynthesis. To affect the rate, we have to change something that is required. Well, one, we can change the amount of CO2. Excuse me, I'm so sorry. We can change the amount of water, but in this lab, the amount of water is not something that we can change, but it is a factor that can affect the rate of photosynthesis. We can change the intensity of light. And another thing that we can change is the temperature. Because remember, if things get too hot, then enzymes denature and reactions don't occur. If they're too cold, the molecules don't move fast enough and reactions tend to slow down. We can also change the color of the light, and that falls into the intensity of light as well. So all of these are things that we can change. And in class, each group is deciding which independent variable they're going to change. If you're at home, you'll have a few options on the virtual lab, but not nearly as many. Just choose one, collect your data, and complete the lab that way. All right, well, if y'all have any questions about photosynthesis, let me know, and I will respond to any emails that you send or questions that you post on Google Classroom. So reminder, you have a quiz tomorrow on photosynthesis diagram and equation, and also be working on your lab graph and the follow-up questions that will be attached to it. They'll be linked separately on Google Classroom, but please make sure that you finish those. And tomorrow we will wrap up photosynthesis after the quiz, and we'll start talking about cellular respiration. I hope y'all have a great evening. Bye.